नमस्कार, आदाब, सत श्री अकाल दोस्तों. Today we are going to start the molecular biology of genes. In this chapter, we are going to begin with the replication of DNA. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. The semi-conservative model for DNA replication was given by Watson and Crick, लेकिन एक्सपेरिमेंटली उसका प्रूफ दिया है मेजल्सन एंड स्टाल ने इन द प्रोकैरियोट्स ई कोलाई. They grew E. coli in a medium containing NH4Cl using N15, a heavy isotope of nitrogen, as the only source for many generations. इसका नतीजा ये हुआ, N15 was incorporated into newly synthesized DNA. The heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride density gradient. N15 is not a radioactive isotope and it can be separated from N14 only based on densities. इसके बाद उन्होंने उस कोश को नॉर्मल N14 सैंपल में ट्रांसफ़र किया. उसके बाद जो देखा गया, they used the similar technique. The various samples were separated independently using cesium chloride gradients to measure the densities of DNA. Then they found out the DNA was extracted from the culture one generation after the transfer from N15 to N14 medium. After 20 minutes, E. coli had a hybrid or immediate density having N15 and N14 both. Now we talk about the machinery and the enzymes required for DNA replication. There is a specific definite region in DNA where replication occurs. This region is called the Ori site. Prokaryotes and viruses have only single Ori site, whereas eukaryotes have several Ori sites. The unit of DNA or a single piece of DNA that can initiate replication is known as replicon. There are various enzymes involved in DNA replication. Number one is हेलिकेज. अब ये हेलिकेज क्या करती है? हेलिकेज डीएनए का डबल हेलिक्स सेपरेट करती है, उनको अलग करती है. The unwinding of double-stranded DNA creates single-stranded DNA which is stabilized by single-strand binding protein which actually prevents the reformation of hydrogen bonds between the separated DNA strands. After the formation of single-stranded DNA, a new daughter strand is to be formed opposite to the parental strands. During replication, one of the daughter strand is synthesized continuously, which is known as leading strand, whereas the other daughter strand is synthesized discontinuously, called lagging strand. The lagging strand is made up of small fragments with 1000 to 4000 nucleotide. This is known as Okazaki fragment. The DNA polymerases catalyze the polymerization only in one direction, that is from 5' to 3' only. For this reason, the replication is also known as semi-discontinuous replication. RNA primers are finally removed By 5' to 3' exonuclease activity of DNA polymerase 1, and a gap is created by removal of RNA primer. Now this is also filled by DNA polymerase 1. Finally, the Okazaki fragments are joined by using DNA ligase. Thank you and subscribe to my channel